So hello everyone, welcome. So this time we're going to talk about chapter 24, the genetic basis of development. So basically what we are trying to see here is to how an organism from a zygote after fertilization turns into a complex individual or its adult stage. We will also try to look into how cells know how to rearrange and how to become what they are supposed to be. In this chapter, we will introduce to you some of the basic mechanisms that control development for plants and animals, focusing on what has been learned in molecular and genetic studies. After introducing the basic molecular processes underlying development, we will also consider as to how cells become different from each other and the factors that establish spatial pattern of these different types of cell in embryo. Next, we'll also examine in more detail the molecular basis of developmental phenomena as examples of the general principles of development. Finally, we'll also discuss about how researchers can learn about evolution by comparing the developmental processes of different organisms. So this is going to be an interesting chapter. So this time we're going to have a quick overview about how an organism from a zygote turns into a full individual. So you can see there on your screen in the upper right, it is basically eggs from a frog, which is turned into a tadpole. It is very complex. So we're gonna discuss how is it going to happen. So first things first, one of the things that happen for an organism to grow is cell division. And this occurs through the process of mitosis. Because remember, meiosis is important for generating haploid gametes. But for an organism to grow and build mass, it must first undergo mitosis. And next, now once you have these cells proliferating and you make more cells, some of the cells we're gonna start to differentiate, to develop specialized structures and functions so that the organism will work correctly. We will also unlock this term, the morphogenesis, because organisms are not just balls of differentiated cells. It must have a three-dimensional shape. So you have to have a patterning information for 3 dimensions. This is a very complicated task. So the big question right here is, how is it going to happen? The answer is all through differential gene expression. So from you, a zygote turns into you, a complex entity, all contained in the DNA of your genome. So that's pretty amazing. So this time we're going to talk about model organisms. So the organisms chosen for understanding broad biological principles is called model organisms. Among the different favorite model organisms include Drosophila melanogaster or the fruit fly, the nematode, the Caenorhabditis elegans, the mouse, the Mus musculus, the zebrafish, the Danaeorareo, and the plant, the Arabidopsis thaliana. So the big question right here is, what are some of the important criteria for model organisms chosen for the study of developmental genetics? So the important criteria include easily observable embryos, short generation times, high reproductive rates, be easy to grow in the lab, a sequenced genome, and the other pre-existing knowledge based on their genetics and biology. So on the preceding slides, I will acquaint you one by one about these model organisms. The first model organism would be the fruit fly or the Drosophila melanogaster. The fruit fly was the first model organism chosen by Thomas Hunt Morgan and intensively studied by other generation of geneticists after him. So the fruit fly is small and easily observed in the lab, and it has a generation time of only 2 weeks and produces many offspring, and embryos grow outside the mother's body. In addition, vast amount of information about its genes and other aspects of biology is presented. However, because first rounds of mitosis occurs without cytokinesis, parts of its development is superficially quite different from what is seen in other organisms. The next model organism would be the nematode or the Caenorhabditis elegans. So the nematode normally grows in the soil, but it is easily grown in petri dishes. So its features include all of this. So it is only millimeter long, simple, transparent body with only few cell types. And it grows from a zygote to a mature adult for only 3.5 days. It has also a sequence of genome And because individuals are hermaphrodites, meaning it is easy to detect recessive mutations. So for the information of everyone, hermaphrodites are organisms that has a complete or partial reproductive organ that produces gametes normally associated with both male and female sexes. And it has a self-fertilization of heterozygotes, produces homozygous recessive um, offspring with mutant phenotypes. So the next model organism would be the mice or the mus musculus. So it has a long history as mammalian model development. Much is known about its biology, including its genes. So genome and genes are roughly the same as humans. So researchers are adept of manipulating mouse genes that transform into transgenic mice or genetically modified mice, and mice in which particular genes are knocked out by mutations. Its generation time is only 9 weeks. However, genes are complex animals with large embryos, and its embryos develop inside the mother's uterus, which is hidden from view, which is the disadvantage of this model. Unlike the Drosophila melanogaster, which is its embryo develop outside the mother's body. So the second to the last model organism would be the zebrafish, the Danio. So the zebrafish has some of its unique advantages. So these small fish, only 2 to 4 centimeters long, are very easy to grow in the lab in large numbers. And it has a transparent embryo that develops outside the mother's body. So although generation time is about 2 to 4 months, the early stages of development proceeds quickly. By 24 hours after fertilization, most of the tissues and early versions of organs are already formed. After 2 days, the fish hatches out of the egg case. Studying the molecular genetics of plant development, scientists are focusing on the small weed, the Arabidopsis thaliana, which is a member of the mustard family. So we can see here all of its features. So one plant can grow thousands of progeny after 8 to 10 weeks. It is a hermaphrodite, meaning each flower consists of ova and sperm. And for gene manipulation research, scientists induce cultured cell to take up foreign DNA. It is called the genetic transformation. Its relatively small genome, which consists of 100 million nucleotide pairs, has already been sequenced. So these developmental principles we have learned throughout this will be presented through this chapter. So stay tuned.
The first topic for chapter 21 is cell division, cell differentiation, and morphogenesis. In the embryonic development of most organisms, the zygote gives rise to many differentiated and specialized cells, in which these differentiated and specialized cells are grouped depending on what function it has to do with the body. For example, in animals, muscle cells enables animals to move. Nerve cells transmit signals to muscle cells for muscle cells to respond to certain stimuli. So these differentiated cells are then grouped into tissues, then tissues into organs, then organs into organ system until it can create a certain organism. So this development from cell to organism is done by 3 processes, 3 interrelated processes, which are cell division, cell differentiation, and morphogenesis. Process is cell division. In cell division, the zygote gives rise to a large number of cells, in which the parent cell gives rise to another daughter cells, depending on what type of cell division it may be. But cell division alone cannot make a fully functional organism. It needs another process, namely cell differentiation. In embryonic development, the cell does not only increase its number, but the cell also undergo cell differentiation. So cell differentiation happens when cell is specialized in structure and in function. An example of cell differentiated in plant is called the mesophyll. So the mesophyll is responsible for the photosynthesis of plants. So cell that has been differentiated are not randomly distributed. Moreover, cells that has been differentiated are grouped into organs and organ system. So, the physical properties that gives an organism its shape is caused by morphogenesis. So, the cell division, the cell differentiation, and morphogenesis overlap over time. Morphogenetic events happen early in the embryonic development, while the cell division and cell differentiation is still happening. Cell differentiation, cell division, and morphogenesis plays an important role in the embryonic development both in plants and animals. However, morphogenesis, both in plants and animals, have great difference, or they have significant differences. In animals, but not in plants, movements of cells and tissues is important for embryonic development. This movement gives the three-dimensional shape of an organism. Let's take, for example, the starfish. So the starfish started in an embryo, which cell divides until it creates a hollow form of cells, which is called the blastula. Then this blastula then reorganizes, then recreates its linings until it creates the gastrula. Then this gastrula then cell divides depending on what lining it is. Then it creates the fully abled functional starfish. In plants, but not in animals, morphogenesis and growth of the overall size in plants is not restricted only at the embryonic level, but rather the growth of the overall size of plants is done throughout its lifetime. It is because plants continually develop or grow new organs, which is the site for embryonic development. And that organ is called the apical meristem, which are found in the shoot and tip of every plant. During cell division and morphogenesis, the behavior and function of the cell is very much different from each other, even though they are of the same cell and they are of the same cycle. In the next chapter, you will know why this thing is happening.
I am here to talk about different cell types result from differential gene expression in cells with the same DNA. Difference between cells in multicellular comes almost entirely from differences in gene expression, but not from differences in the cell's genome. Genomic equivalence means all the cells of an organism have the same genes. One experimental approach for testing genomic equivalence is to see whether a differentiated cell can regenerate a whole organism. During 1950s, Stewart and his students performed an experiment at Cornell University. They are working with carrot plants. I have here a figure about their experiments. They found out that differentiated cells taken from the roots of the carrots could grow into normal adult plants, each genetically identical to the parent plant. Therefore, this shows that differentiation does not necessarily involve irreversible changes in the DNA. Totipotent cell means cell is capable of generating a complete new organism. But using one or more somatic cells from a multicellular organism to make another genetically identical individual is called cloning, and each new individual made in this way called clone. In animals, differentiated cells generally do not divide in culture. Therefore, animal research had to use a different approach to question of whether differentiated animal cells can be totipotent. Their approach was nuclear transplantation. Researchers conducted an experiment on frogs. They transplanted a nucleus from an embryonic or tadpole cell into a nucleated egg of the same species. From these results, researchers concluded that something in the nucleus does not change as animals differentiate. Various cell types of the body of an animal differ in structure and function, not because they contain different genes, but because they express different set of genes from a common gene. Since 1997, cloning has been demonstrated in numerous mammals, including mice, cats, cows, horses, pigs, and lambs. In most cases, the goal has been to produce new individuals, this known as reproductive cloning. Dolly is the first lamb clone from adult sheep. Analysis showed that Dolly's chromosomal DNA was indeed identical to that of the nucleus donor, but at the age of 6, she suffered complications. Dolly's premature death led to speculation that her cells were more older than those of a normal sheep, possibly reflecting incomplete reprogramming of the original transplanted nucleus. Another example is the first cloned cat, named Copycat. She has a calico coat like her single female parent, but the color and pattern are different due to random X chromosome inactivation, which is a normal occurrence during embryonic development. Many cloned animals exhibit various defects. For instance, cloned mice are prone to obesity, pneumonia, liver failure, and premature death. Scientists believe that even cloned animals that appear normal are likely to have subtle defects. Further support for the idea that cells became different by expressing different sets of genes from the same genomes come from the study of a group of cells called stem cells. Stem cells are able both to replenish their own population and to generate cells that travel down various differentiation pathways. I have here a figure of animal stem cells, which can be isolated from early embryos or adult tissues and grown in culture. Stem cells are self-perpetuating, relatively undifferentiated cells. Embryonic stem cells are easier to grow than adult stem cells and can theoretically give rise to all types of cells. The range of cell types that can arise from adult stem cells is not yet fully understood. The evidence that we've discussed shows that nearly all the cells in a plant or animal contains the same set of genes. Next, we'll look at the main processes that give rise to different cell types, that is, the molecular basis of cell differentiation.
So now, I'll be discussing to you the transcriptional regulation of gene expression during development. For example, as a result of transcriptional regulation, liver cells are specialized in making albumin and lens cells are specialized in making crystallines. Although the chromatin cells appear to be unchanged under the microscope, determination has occurred, and they are now myoblasts. Myoblasts start to churn out large amounts of muscle-specific proteins and fuse to form mature, elongated, multinucleate skeletal muscle cells. This gene encodes a myosin protein, a transcription factor that binds to special control elements in the enhancers to various target genes and stimulates their expression. Some target genes of MYOD encode still other muscle-specific transcription factors. MYOD also stimulates expression in the MYOD gene itself, thus perpetuating its effect on maintaining the cell's differentiated state. The secondary transcription factor activates genes for proteins such as myosin and actin that confer the unique properties of skeletal muscle cells. These observable changes are actually the outcome of the cell's developmental history extending to the first mitotic division of a zygote. At the end of this process, an embryonic cell is irreversibly committed to its final fate. It is said to be determined. If a committed cell is experimentally placed on another location inside the embryo, the cell will still differentiate to its normal fate. These proteins are found only on a specific cell type, and it gives cell characteristic structure and function. The first evidence of differentiation is the appearance of mRNAs to these proteins. In most cases, the pattern of gene expression is controlled by the level of transcription.
So today, I am going to discuss about the cytoplasmic determinants and cell-to-cell signals in cell differentiation. So there are two sources of developmental information that tells a cell which gene is to be expressed at that given time. First is the cytoplasm of the egg cell. The second one is the environment around the cell. The cytoplasm of the egg cell contains RNA, mRNA, and protein molecules that helps determine the developmental fate of the embryo, while the environment around the cell allows the passing of signals to change the gene expression of the receiving cell. So, when we talk about the cytoplasm of the egg cell, we are considering the cytoplasmic determinants that regulate the gene expression in the embryo or zygote, and then it affects the developmental fate of the said zygote. So, I have here a figure of the cytoplasmic determinants in the egg cell. Let us first focus on the egg cell. Notice that in this figure, we have the green triangle and the orange circle that represents molecules in the cytoplasm of the egg cell. The cytoplasm of the egg cell is not homogeneous, since it is composed of RNA, mRNA, and protein molecules that is encoded by the mother's DNA. It is also unevenly distributed around the cell. The heterogeneity of the egg cytoplasm creates an impact on the developmental fate of an embryo. These maternal substances such as RNA, mRNA, and protein molecules that influences the course of early development in the embryo or the cell are called cytoplasmic determinants. These substances regulate the gene expression that can also affect the developmental fate of an embryo. During fertilization, when the sperm cell and the egg cell unite, only the nucleus of the sperm cell enters the egg cell. Thus, we can conclude that the other factor that contributes to the developmental fate of an embryo is the cytoplasm of the egg cell. So after fertilization, mitosis occurs, and it produces two daughter cells. Notice that in this figure, the cytoplasmic determinants or the substances around the cytoplasm of the cells are different from each other. The two daughter cells are now exposed to different cytoplasmic environments, and this contributes to what kind of cell it is going to be. The set of cytoplasmic determinants that a cell receives helps determine the developmental fate of an embryo by regulating the gene expression of the cell's gene during the course of cell differentiation. The environment around the cell affects the developmental fate of an embryo through the process of induction. Signal molecules from embryonic cells cause transcriptional changes in nearby target cells. So, I have here a figure of the induction of 32 cells. When we enlarge the bottom part of these neighboring cells, we can see the image below. So let us first analyze the cell at the bottom. This cell transfers signaling molecule, also called induced molecules, to the other cell. Let us consider that this cell is already differentiated and its fate is already determined. The signal receptor in blue receives the signal molecule from the differentiated cell. The signal molecule then travels to the signal transduction pathway to reach the nucleus. When the signal molecule or the induced molecule reaches the nucleus, the gene expression of that cell is changed. This happens to facilitate cell differentiation of neighboring cells to function as a whole. In animals, induction happens by contact with cell surface molecules on neighboring cells and the binding of growth factors secreted by neighboring cells. In plants, plasmodesmata allows the induction process. Plasmodesmata allows signal to pass from one cell to another. The synthesis of these signals is controlled by the embryo's own genes. So induction is the process wherein molecules cause changes in nearby target cells. This is very important as a source of developmental information when the number of the cells increase. Thus, the interaction between embryonic cells induce differentiation of the many specialized cell types, making up a new organism.
Morphogenesis shapes an animal's body or a plant's body. But before morphogenesis can shape it, there should be an established body plan. Okay, just like a building. Before you can stack up all your hollow blocks, pour all your cements, there should be a blueprint of your building. So, a body plan is important for morphogenesis. So, a body plan is also influenced by what we call cytoplasmic determinants that are found in the cytoplasm of cells. So we have what we call pattern formation, the development of a spatial organization in which the tissues and organs of one organism are all in their characteristic places. So it's like a distribution or your initial deployment to your assigned area or field until you got your permanent position. In plants, pattern formation continually occurs in the apical meristem. So it's like the apical meristem is one of the busiest parts of a plant. In animals, pattern formation is mostly limited to embryo stage or the juvenile stage, except for those species who can regenerate their lost parts, because that means another pattern formation. Okay, so I have here a picture of an early embryo development. By the way, pattern formation starts at early embryo, so I'm showing there too. Okay, let's have an example. Okay, let's go with a building. Okay, before you can start your building construction, of course you need to locate your front, your back, and the sides of your building, right? So the same goes with the specialized tissues, your organs of the animal. They can't appear unless your location of your head, the tail, the sides, the front, and back are determined. So, okay, just think of it. So in our embryo, the germ layers appear. And we know that our organs, most of us, our body are derived, parts of our body are derived from the germ layers. So we have here a picture of cytoplasmic determinants in the egg. So as we can see, there are green triangles and orange circles. So those green and orange things, so they stand for the different cytoplasmic determinants. And as we can see, this different cytoplasmic determinants are already there long before the sperm comes to the egg to fertilize. And now that our egg is now fertilized, it becomes a zygote. And as we can see that the green triangles and the orange circles are unevenly distributed. Okay, so after fertilization comes the mitotic division. So the nucleus of the zygote, the nuclei of the zygote, they become exposed to different sets of cytoplasmic determinants, which results to expression of different genes. There are things called molecular cues. So these molecular cues, they control the pattern formation, and collectively they are called positional formation. So positional formation are provided by the cytoplasmic determinants and the inductive signals. Okay, so inductive signals. So this picture is a good illustration of induction by nearby cells. So we have here an early embryo with already 32 cells. Yes, and 2 of these 32 cells are having communication. Okay, so the bottom cell is sending chemical chemicals to the other cell, signaling it to change its gene expression. Okay, so as you can see, those orange circles, so they are the chemical signals sent by the lower cell that are received by the receptors of the cells up there. Okay, so I think it's much easier to compare it to your neighborhood. It's like neighbor one is telling the neighbor, like, hey neighbor, can you lower your music because I can't concentrate here. So Drosophila, fruit flies. So Drosophila have modular construction. It is an ordered series of segments. So these segments, they make up the three body parts: the head, the thorax. Well, thorax, it includes the mid-body from which the wings, the legs extend, and the last one is the abdomen. Okay, the head, the thorax, and the abdomen. Okay, just like other bilaterally symmetrical animals, the Drosophila, the fruit fly, it has also a posterior axis and a dorsal ventral axis. In Drosophila, cytoplasmic determinants are localized in the not yet fertilized egg cell. And these cytoplasmic determinants provide the positional information of these two axes, the posterior axis and the dorsal ventral axis, even before fertilization occurs. Drosophila egg cell develops in the female's ovary, and nerve cells and follicle cells supply that egg cell with the substances it needs for development. And that nerve cells, along the way, it will shrink and disappear. And the egg is now fertilized. So after fertilization and laying of the egg, the following events follow. First 10 mitotic divisions happens very quickly, and it is just composed of S and M phases. Therefore, the number of cytoplasm doesn't change. It's still one cytoplasm, and there's no cytokinesis. Therefore, our embryo is one big multinucleate cell. In the 10th nuclear division, the nuclei begins to migrate to the periphery of the embryo, forming a blastula-like stage, and we call it blastoderm. Then the plasma membrane develops each nuclei into separated cells. And the body plan at this point, I am referring to the two axes. The body plan is already determined by this time, and there is that yellow thing, that central yolk is nourishing the whole embryo, and the eggshell continues to protect it. The subsequent events in the embryo creates clearly visible segments which at first look very much alike. Some cells move to new positions, and there are organs started to grow, and there is a worm-like larva hatching out of the shell. And the Drosophila happens to have three larval stages from which it eats, it grows, and molts or shed its tough outer layer. And the third larval stage of Drosophila becomes enclosed in a case, thus forming the pupa. And then it's metamorphosis, the change from larva to adult fly. It happens inside the pupa, and right after that, the fly now emerges.
Hello! You've already been acquainted with the life cycle of a Drosophila. Moving forward, let us look back in the past and see how scientists have unlocked the mechanism of development that enables us to see a better picture on how such mechanism works in the molecular level, as I discussed to you the genetic analysis of early development. Classical embryologists during the first half of the 20th century made a detailed anatomical observation on the embryonic development of a number of species and performed varied experiments wherein they manipulated its embryonic tissues. Although these experiments provided and laid down the groundworks for the better understanding on the mechanism of development, it did not provide the specific molecules responsible for such development or the information on how certain patterns on certain organisms are established, therefore creating a gap of information. However, during the year 1940s, a visionary American biologist by the name of Edward B. Lewis filled this gap and showed to everyone that the genetic approach can be used as a tool to investigate the development of a fruit fly. Lewis studied bizarre mutant flies with physical developmental defects and discovered the gene responsible for the control pattern formation of the fly's late embryonic development. This research provided the first concrete evidence that the genes somehow direct these developmental processes that embryologists have been studying for many years. On the other side of the coin, German researchers Christian Noslin-Volhard and Eric Yeschhaus provided insights on the pattern formation of the Drosophila in its early embryonic development. However, their study faced three major problems. First, the Drosophila has about 13,700 genes, and only a few of these genes are responsible for the segmentation. That's why finding it would be very, very difficult. Second, the mutants that affect segmentation are likely to be embryonic lethal. So flies with embryonic lethal genes will never reproduce. That's why breeding it for study is impossible. And lastly, the maternal effect on the axis formation will require the researchers to study the maternal genes as well. That's what it makes this research more complicated, longer, and almost impossible to do. To get around these problems, these two smart researchers focus their study in the recessive mutation that can be propagated to form heterozygous flies. Findings identified 1,200 genes essential for embryonic development, 120 of which are essential for pattern formation that can lead to normal segmentation. Years after, they were able to group the genes by general function, map them, and clone many of them. Lewis's early work and these two German researchers' study provided a coherent picture of the Drosophila's development. And in 1995, Noslin-Volhard, Weishaupt, and Lewis were awarded with the Nobel Prize. At this juncture, I am now going to discuss about axis formation, segmentation patterns, and the identity of the Drosophila body parts, and explain how these processes occurs as the fly's embryo develop. Now, let us first tackle about the axis formation. So the initial establishment of the axis of the Drosophila's body is made possible by the cytoplasmic determinants. These cytoplasmic determinants are encoded in a gene named maternal effect gene. The maternal effect gene is a gene that when mutants in the mother produces mutants in the phenotype of its offsprings, regardless of its own genotype. And these maternal effect genes are also called egg polarity genes, and these egg polarity genes will determine the orientation or the polarity of the fly's body. Posterior anterior axis as well as the ventral dorsal axis will be determined by different sets of maternal effect genes. To understand better how the maternal effect gene determines the formation of the body axis of the offsprings, let us focus our discussion to a gene called the bicoid gene. Bicoid means two-tailed. So in this case, the developing embryo of the larva lacks its first half of the body, having posterior structures at both sides, just like what the image shows here. In the image, there is a wild type larva and a mutant larva. The wild type larva has no mutation at its maternal bicoid gene. Why? Because both posterior and anterior structures are present at its respective places. The mutant larva, on the other hand, has its thoracic and abdominal segments at both sides, as what referred by the numbers. So this signifies that this larva has a mutation at its maternal bicoid gene. This discovery suggested to the researchers that the product of the maternal effect gene is essential for the formation of the anterior structures of the fly, and as well as the idea that these bicoid genes are concentrated at the anterior end. Based on this idea, the researchers formulated a hypothesis that a gradient of substances called morphogens are responsible for the formation of the embryo's axis and the features of its form. To prove this hypothesis, researchers used DNA technology and modern biochemical methods. And as predicted, they found out that bicoid mRNA are highly concentrated at the extreme anterior end of the mature egg cell. After the egg cell is fertilized, the mRNA is translated into protein. The bicoid protein will then diffuse from the anterior end towards the posterior end, having of course a high concentration of it at the anterior end. Bicoid protein is only one of the several morphogens involved in axis specification. So the research of bicoid is very important. Why? Mainly because, first, it helped identify a specific protein required for one of the most earliest steps in pattern formation. Second, it helped us understand the critical role of the mother in the initial phases of embryonic development. And lastly, it helped prove that the gradient of molecules is a key developmental concept in a number of species. You already know the mechanism of the axis formation of a Drosophila's body. Now, I am going to discuss about the segment formation. So the difference between the segment formation and the axis formation is that in the axis formation, the four major axes of the Drosophila's body is being formed. These are the posterior anterior axis and the dorsal ventral axis. In the segment formation, however, it is a process wherein the fly's body is being divided into segments, wherein at the later part will make up the three major body parts of the Drosophila, which is the head, the thorax, and the abdomen segment. Segmentation pattern, on the other hand, is made possible as the production of protein in the segmentation gene initiates. So after the four major axes are formed, the segmentation gene will then be activated. A sequential activation of three sets of segmentation genes provides the positional information for the increasingly fine details of the animal's modular body plan. This three sets of genes are the gap genes, the pair rule genes, and the segment polarity genes. The first gene to be activated is the gap gene. Gap genes map out the basic subdivisions along the posterior-anterior axes. Mutation in this gene will cause gaps in segmentation. The second gene to be activated is the pair rule gene. Pair rule genes define modular pattern in terms of pairs of segments. Mutation in this gene will result with an embryo having half the normal number of segments. The third and final gene to be activated in segment formation is the segment polarity gene. The segment polarity gene sets the posterior anterior axes of each segment. Mutation in this gene will have a normal number of segments in the embryo, but with part of each segment being replaced with a mirror image repetition of some other parts. After the formation of the four major body axes of the Drosophila, followed by segments being established, the next gene to be expressed will determine the specific anatomy of each segment along the length of the embryo. This gene is called the hemiotic gene. The hemiotic gene, also known as the master regulatory gene, was discovered by Edward B. Lewis. He defined this gene as the gene responsible for the late embryonic development of the Drosophila. The hemiotic gene will specify the type of appendages and other structures that will form in a certain segment. These structures include the antenna, the legs, the wings as well, and many other appendages and structure that is appropriate to form in a certain segment. Now, mutation in this hemiotic gene will cause physical developmental defects to the Drosophila. It can cause the entire structure characteristic of a specific segment of the animal to rise on a wrong segment, causing this kind of result shown in the micrograph. The micrograph compares a mutant fruit fly to a wild-type fruit fly. The wild-type fruit fly has a pair of antennae on its head, while the mutant fruit fly has a pair of legs instead of an antenna on its head. What happens here is that there was a mutation in the thoracic gene of the Drosophila. That's why instead of an antenna, a pair of legs is seen. Without the mutation, this hemiotic gene will complete the development of the Drosophila and its different parts of the body. To sum up everything, this image shows the hierarchy of gene activity on the early development of the embryo of a Drosophila. The axis formation is made possible as the maternal effect gene was activated. So after the formation of the four major body axes, segmentation follows. In segment formation, there is a sequential activation of three sets of segmentation genes, namely the gap gene, followed by the pair rule gene, and lastly, the segment polarity gene. After the segments were established, the hemiotic gene will work and specify the anatomy present in each segment. So there is the formation of the antenna in the head, the legs, and the wings. So basically, this processes will sum up the early development of a Drosophila.
Today, we are going to discuss about cellular elegans and the role of cell signaling. The development of a multicellular organism requires close communication between cells. This communication already occurs even before fertilization. So once the embryo is truly a multicellular, with membranes enclosing each individual nucleus and cytoplasm, inductive signaling between the embryo's own cell becomes increasingly important. As we've seen, the ultimate basis for the differences between cells is transcriptional regulation, which is the turning on and off of specific genes. On the other hand, it is induction, the signaling from one group of cells to the other, that brings about differentiation. In some cases, cell signaling also leads to the program death of specific cells, a phenomenon which is also crucial to normal embryonic development. The nematode Caenorhabditis elegans is used as a model organism for investigating the role of signaling, induction, and program death of specific cells. So the transparency of its embryo allows the researchers to trace the lineage of every cell, from zygote to its own adult. The diagram shows a detailed lineage only for the intestine, colored yellow, which is exclusively derived from one of the first 4 cells formed from the zygote. The intestinal cell lineage does not happen to include any program cell death. So the large white cells are eggs, which will be fertilized internally and released through the vulva. As early as the 4-cell stage in C. elegans, cell signaling helps direct daughter cells down to their appropriate pathways. For instance, as shown in figure 21.16a, a signal from cell 4 acts on cell 3 so that the daughter cells of cell 3 will eventually give rise to the intestine. So the signal is a cell surface protein made by cell 4, which can be recognized and bound to the cell surface receptor protein on cell 3. This interaction triggers events inside cell 3 that result in one end of the cell, which is the posterior end, and the other becoming different. So when cell 3 divides, the posterior end will eventually go on to make the intestine, while the anterior end will make a different phase. If cell 4 is experimentally removed in the early 4-cell stage of development, then no intestine is developed. But if cell 3 and cell 4 are recombined, then the intestine will develop as normal. These results help researchers to know the role of induction in the early nematode development. Induction is also crucial later in the nematode development as the embryo passes through 3 larval stages prior to becoming an adult. As you can see in figure 21.16b, the vulva, the tiny opening through which a worm lays its egg, arises from 6 cells that are present on the ventral surface at the second larval stage. The anchor cell in the gonad secretes many copies of a particular signal protein, providing a strong inductive signal precursor cell, the dark blue one, causing it to form the inner part of the vulva. The two adjacent cells, the medium blue color, receives weaker signal and are induced to form the outer vulva. The three remaining precursor cells, the light blue one, are too far away to receive the signal. That's why they became part of the worm's epidermis. These two examples of induction during nematode development illustrates a number of important concepts that apply elsewhere in the development of C. elegans and many other animals.
First, in the developing embryo, sequential inductions drive the formation of organs. Second, the effect of an inducer can depend on its concentration, just as we saw in cytoplasmic determinants in Drosophila. Third, inducers produce their effects via signal transduction pathways similar to those operating in adult cells. And lastly, the induced cell's response is often the activation or inactivation of genes, transcriptional regulation, which in turn establishes the pattern of gene activity characteristics of a particular kind of differentiated cell.
Genetic analysis of C. elegans has underscored another outcome of cell signaling that is also crucial in animal development, the programmed cell death or apoptosis. The timely suicide of cells occurs exactly 131 times in the course of normal development in C. elegans. So in worms and other species, apoptosis is triggered by signals that activate a cascade of suicide proteins in the cells destined to die. During apoptosis, a cell shrinks and becomes low, called lemming. The nucleus condenses and the DNA is fragmented. So in figure A is a normal white blood cell, while figure B is a cell that undergoing apoptosis, and as you can see, it's shrinking and forms webs. So which eventually shared as one cell membrane that is fragmented. The genetic screening of C. elegans has revealed two apoptosis genes, SED3 and SED4. SED stands for cell death, which encodes proteins essential for apoptosis. Three proteins, SED3, SED4, and SED9, are critical to apoptosis and its regulation in the nematode. SED9 serves as a master regulator of apoptosis, and it acts as a brake in the absence of a signal promoting apoptosis. So, as long as SED9 is active, the cell remains alive. On the other hand, when a cell receives a death signal, SED3 is inactivated, relieving the inhibition of SED3 and SED4. So active SED3 triggers a cascade of reactions leading to activation of nucleases and proteases. So the main proteases of apoptosis of a nematode is called capsaicin. So the action of these enzymes causes the changes seen in apoptotic cells and eventually cell death. In vertebrates, apoptosis is essential for the normal development of the nervous system, for the normal operation of the immune system, and for the normal morphogenesis of hands and feet in humans and paws in other mammals. Figure 21.19 shows the effect of apoptosis during pod development in humans and mouse. So, in mouse, humans, and other mammals, as well as land birds, the embryonic region that develops into feet or hands initially has a solid platelet-like structure. Apoptosis eliminates the cells in the interdigital regions, thus forming the digits. So the embryonic mouse paws shown here are staining so that the cells undergoing apoptosis appear light green. So apoptosis of cells begins at the margin of each interdigital region. So this left picture then peaks as the tissue in these regions is reduced, so the middle picture and is no longer visible when the interdigital tissue has been eliminated, so the right picture. Behind the discovery of cell signaling in C. elegans are Sidney Brunner, John E. Sulston, and H. Robert Horvitz. So the role signaling was discovered less than 30 years ago. The importance of their study was highlighted in 2003. They won a Nobel Prize for medicine for they advanced the understanding of how genes regulate organ growth and the process of growth cell death.
Plant development, cell signaling, and transcription regulation. The genetic analysis of plant development using models such as Arabidopsis has lagged behind that of animal models simply because there are fewer researchers working on plants. For example, in 2000, when the DNA sequence of Arabidopsis had been completed, fewer than 5% of its genes had been defined by using mutational analysis, whereas over 25% of the genes of both Drosophila and C. elegans had been identified using the same way. Now let's go on to the mechanism of plant development. Cell lineage is much less important for pattern formation in plants than in animals. Many of the plant cells are totipotent, meaning their fates depend on the positional information than in cell lineage. The major mechanism regulating the development are cell signaling and transcriptional regulation. Embryonic development of most plants occurs inside a seed and thus inaccessible to study. A fully mature seed contains already the fully formed embryo, and thus there are other important aspects of plant development that can be observable throughout the plant's life, and its meristem, particularly in the apical meristem where the cell division, morphogenesis, and cell differentiation give rise to new organs such as the leaves and the flowers. And now we're going to discuss about the pattern formation in floral meristem. Floral meristem is the apical meristem where flowers are produced. Pattern formation of flowers. Environmental signals such as day length and temperature trigger signal transduction pathways that convert ordinary shoot meristem to floral meristem, causing a plant to flower. The researchers have combined a genetic approach with tissue transplantation to study induction of the development of tomato flowers. As shown in the figure, floral meristem is a bulb consisting of 3 layers. All these 3 layers participate in the formation of a flower. A flower is a reproductive structure consisting of 4 types of organs, which are carpels, which contains the egg, the stamen, which contains the sperm-bearing pollen, the petals, and the sepals, which is a leaf-like structure outside the petals. Now, in a mature plant, all these types of organs are arranged radially rather than linearly like the body structure of the Drosophila. Tomato plants homozygous for a mutant allele called fasciated produce floral organs with abnormally large number. So to study what controls the number of organs, researchers performed the grafting experiment. They grafted stems from the mutated plants onto the wild-type plants, and then they grew the plants from the shoot that sprouted near the graft sites. Many of the new plants were chimeras. Chimeras were organisms that mixture with genetically different cells. Some of these chimeras produce floral meristem with which 3 layers did not come from the same parent. So in order to determine the parental source, researchers monitor other genetic markers such as monitoring the unrelated mutation causing yellow leaves. In contrast with the gene controlling organ number are the genes controlling organ identity. It determines what type of structure will grow from the meristem. Like for instance, whether that particular outgrowth from the floral meristems becomes flowers or leaves.
So much of how we knew or much of what we know about such organ identity genes comes from the research on floral development in Arabidopsis. Organ identity genes are similar or analogous with that homeotic genes in animals and are often referred to as plants' homeotic genes. In a plant with homeotic mutation, specific organs are missing or repeated. So in this figure, it shows the floral identity genes mutations. So a wild type Arabidopsis has 4 sepals, 4 petals, 6 stamens, and 2 carpels. If organ identity genes called apitala is mutated, the identities of these organs in the 4 walls are carpels, stamens, carpels, no sepals, and no petals. So clearly, the mechanisms on the development of the plants are clearly the same with the development of the animals as being discussed earlier.
So, concept 21.4 is about the comparative studies which help explain how the evolution of development leads to morphological diversity. So, biologists in the field of evolutionary developmental biology compares developmental processes of different multicellular organisms. They wanted to understand how developmental processes and lead to modifications in the organism's features or even lead to new one. So with the advent of molecular techniques and recent flood of genomic information, we begin to realize that organisms with different of related species with different forms may only have minor differences in their gene sequencing or regulation. So, what is a homeobox gene? So, a homeobox gene is a large family of similar genes that direct the formation of many body structures during early embryonic development. So here in figure 21.23, it shows the conservation of homeotic genes in a fruit fly and a mouse. So molecular analysis of homeotic genes in Drosophila has shown a 180 nucleotide sequence called homeobox, which is found very similar or identical to the homeotic genes found in many vertebrates and invertebrates. So as you can see, homeotic genes that control the anterior and posterior structures of the body occur in the same linear sequence on chromosomes in Drosophila and mice. So the each colored band shows a homeotic gene. The color code indicates the parts of the body which are expressed and the adult body regions that result. So related sequences have been found in regulatory genes of much more distantly related eukaryotes, including plants and yeasts and even prokaryotes. So from these similarities, we can say that homeobox DNA sequence evolved very early in the history of life and was conserved in animals and plants. It was unchanged for years. So take note that not all homeobox containing genes are homeotic genes. Some do not directly control the identity of body parts. So most of animals' genes are associated with development. So in Drosophila, homeobox are are present not only in homeotic genes but also in egg polarity gene bicoid, in several of segmentation genes, and in master regulatory gene. So when protein functions as a transcriptional regulator, homeobox encoded homeodomain, a part of a protein, binds to DNA. So proteins with homeodomains regulate development by coordinating the transcriptional batteries of development by um, switching them on or off. So the selective expression of the regulatory gene varying through time and space is central to pattern formation. So how can the same genes be involved in the development of animals whose forms are very different from each other? So current studies have likely answers like small changes in the regulatory gene sequences of a particular regulatory genes or regulatory sequences of a particular genes can lead to a major change in the body form. So in other cases, um, the same genes can direct different developmental processes that um, that can result to um, different body shapes. So I have here a comparison of the animal and plant development. So it was said that the it was said that the common ancestor of the animal and plant was unicellular. So comparison of the developmental mechanisms of plant and animal would show that development was independently invented on each lineage. So although homeobox genes and Madsbox genes existed in the last common ancestor, Madsbox genes plays a very important role in the regulation of plant development but not in animal development, where homeobox genes are important. The cell movement of animal and plant cells also differ, as animal cells are motile, meaning animal tissues can be folded and can be moved against each other, whereas plant cells are positionally fixed. Plant cells are rigid, made up of cellulose, which prevents movement of the plant cells and tissues. So in this final chapter of the genetics unit, you have learned how genetic studies can reveal much about the molecular and cellular mechanisms underlying development. So, the similarities reflect the common ancestry of life on Earth, but the differences are also very crucial, for they have created the huge diversity of organisms that have evolved today.